According to the species under study and the purpose of the experiment, the samplings for gametes collection may involve different steps. Before sampling, it is important to anesthetize the fish properly. Start by catching the fish with a net and identify the animal by reading its tagged ID. Place the fish inside the anesthesia tank, which should be equipped with oxygen or aeration. The type of anesthetic, its concentration, and the time of exposure must be adapted for the species, fish age, and type of procedure you want to do. Turn the fish upside down. If it has lost its ability to return to the upright position, proceed with sampling. These procedures are common for other finfish species, like gilt-head sea bream. Start to put the right volume of the anesthesia in a proper tank. Introduce the fish in the anesthesia tank until it loses locomotor activity control. In the end, place the fish on the sampling bench and start the procedures. In aquaculture, some species reproduce naturally, but others require hormonal treatments. There are several methods, but the most common are implants and injections. Hormonal implants are easy to apply and allow a slow release of the hormone throughout a specific period, which depends on the type of hormone and its concentration. When ordering the hormone, be aware that the dose is calculated according to the fish's weight. To introduce the implant in the fish, insert in a sterilized syringe and perform an intramuscular injection. Apply a topical antiseptic to avoid an infection. In some species, sex identification for gamete collection is easy, but in others you need a microscope. In this lecture, we will focus on sperm collection in different species. In Senegalese sole, during the breeding season, the female gonad increases considerably and it is easy to distinguish males from females. Start by reading the fish ID to be sure that you are sampling a male animal. In sole, the male gonad is located on the fish's right side in the visceral area. Cover the fish head to minimize the stress. Clean the genital area with a neutral solution and a paper tissue. By gently pressuring the testes, collect the sperm samples with a 1 ml syringe. Avoid any contamination by urine. Place the sample in a 1.5 ml tube. Identify it with a fish ID and keep the samples at a low temperature until analysis. In sea bream, sperm samples are collected by abdominal massage. Start by placing the fish in an adequate support and cover the fish head. Clean the genital area as previously described. The stripping must be gentle and performed from the anterior part to the genital area, avoiding any contamination. Sea bream can release a high amount of sperm, so it's better if you use 5 ml tubes to store the samples. Keep it on ice until analysis. In oysters, sex cannot be identified externally, so you need to carefully cut the adductor muscle that holds the two valves. The gonad overlies the digestive tissues, thus clean this area with filtered seawater. For sex identification, perform a small incision in the gonad and observe the content under a microscope with few drops of seawater. Oocytes from females have a round pear shape. Spermatozoa have motility. To collect the sperm, do repeated incisions in the gonad with a sterilized scalpel. Avoid the disruption of the digestive gland and consequent contamination. Collect the sample with a Pasteur pipette and keep it in a proper tube at 4 degrees. Immediately, filter the sperm with 120 micrometer sieves to remove any gonadal tissue debris. Finally, dilute the sample in seawater to have the desirable final concentration for further analysis or fertilization procedures. Sperm motility is the most common technique to evaluate sperm quality. It's easy to perform and directly correlated with fertilization success. Objective motility analysis requires a software. In this example, select the motility feature on your software to evaluate the different motility parameters. Place 10 microliter of seawater in a macular chamber and add 1 microliter of sperm. This will activate sperm movement. After mixing, activate the timer and record motility at each 15 seconds until the end of the movement. Species like Senegalese sole and gilt-head sea bream have short movement duration, but oyster sperm may move for several hours to days. In the end, check your motility data directly in the program or export for further analysis. For blood collection, use a heparinized 1 ml syringe. Blood is collected from the caudal vein in Senegalese sole and gilt-head sea bream. Transfer the blood into a heparinized tube and invert the tube to homogenize with the heparin. The blood samples should be placed directly above the ice until centrifugation. Set the centrifuge for 3000 Gs, 15 minutes and 4 degrees. Place the samples inside and start the process. In the end, transfer the blood plasma, the supernatant, to a different tube and store at -80 degrees until analysis. Several hormones related to reproduction can be measured in this type of sample.